let us see uh, the drug development process, and then let us see where the SAS is used and how the SAS is used. Okay? As I told you already, when it comes to the drug development, whenever any pharmaceutical industry is trying to develop the drug, they will go for a lot of research in order to identify different chemical compounds. According to their requirement, they will do the research and they will search for different chemical compounds. And once they have identified their required chemical compound, then they will think about the synthesis and purification of that particular chemical compound, because it must be cheaper and it must be freely available in the atmosphere. Okay, it must be cheaper and it must be freely available in the environment. Then only they will consider that particular compounds. They will take that particular chemical compound. Why? Because if it is more expensive or if it is not freely available in the environment, it will be a difficult process for developing or producing, okay, drugs. So generally the researchers will prefer, okay, the chemical compounds which are freely available in the environment, and at the same time they must be cheaper for purification process as well as for the synthesis process also. Once they have identified those kind of chemical compounds, they will apply them on the animal cells for the research purpose. They will apply those chemical compounds on the animal cells for the research purpose, and they will do the research in the laboratory conditions. And we will call this as a preclinical trials or non-clinical trials. Whenever they were doing the research on the animal cells, we will call them as preclinical trials or non-clinical trials. Once that particular pharmaceutical industry is satisfied with their research work, then they have to send their research information to the FDA, Food and Drug Administration. Now, FDA will review the investigation of new drug which was sent by the pharmaceutical industry. FDA will review this, and if it was satisfied with the research work, then it will go approval to conduct the clinical trials. Unless the pharmaceutical industry will get the approval from the FDA, it will not conduct the clinical trials. Once it got the approval, then it will proceed with the clinical trials. But directly, the pharmaceutical industry itself will not proceed with the trials. It needs the help of contract research organizations, where they will select the investigator. Here they will recruit the principal investigator. And here only we used to have the clinical research associates. We can say these are nothing but subordinates which we have under the principal investigator. Because generally the clinical trials are conducted according to the guidelines of the principal investigator. He is the decision maker and he will look over the whole clinical trial process. Of course, sometimes he may not go everywhere and he may not take care of the entire clinical trial. So in support of this principal investigator, we used to have some sub-investigators, and we used to have some um, IRB board, institutional review board. Okay, we used to have ethics committee, where they will look over the trial process, and they will give suggestions to the principal investigator so that he can make the decision. Okay, whether he can continue with the trial, or if one particular trial is better than another one, or one chemical compound is better than the other one, or if we got any adverse events. Okay, all that will be given to the principal investigator. All that particular information will be given to the principal investigator, okay, by the institutional IRB or EHC or ethics committee, etc., etc. So based on their views, the principal investigator will decide in what particular pattern he should proceed with the trials. And when it comes to the clinical trial, we know that here we have different phases. When it comes to the phase 1, there the number of When it comes to the phase 1, the main objective is it is used to determine the metabolic and pharmacological actions and maximally tolerated doses. It will give us the information regarding these, or we were looking for the phase 1 to identify this particular information: metabolic and pharmacological actions and maximally tolerated doses. Factors to be identified during the phase 1 is bioavailability, bioequivalence, dose proportionality, metabolism, pharmacodynamics, and pharmacokinetics. Data that we will focus is vital signs, plasma and serum levels, adverse events. Design: it is a single ascending dose types, unblended, uncontrolled. Duration: it will be conducted up to 1 month. Generally, it will be conducted for 1 month. And here, the population or sample is healthy volunteers, persons who are not suffering from any diseases. And the sample size is in between 20 to 80. Once they are succeeded with the phase 1, then they will start conducting the phase 2. Here the objective is evaluate effectiveness and determine the short-term side effects and identify common risks for a specific population and disease. Factors to be identified: bioavailability, drug-to-disease interactions and drug-to-drug interactions, efficacy at various doses, pharmacodynamics and pharmacokinetics, along with the patient safety. Data that they will collect is regarding dose response, intolerance, adverse events, and efficacy. Design: placebo-controlled comparisons, active controlled comparisons, well-defined entry criteria. Of course, it will be conducted for several months, maximum for 1 year. Individuals with target diseases as the population or sample here, and the size is few hundreds, like around 200 to 300. Once they were succeeded with their research work, then they proceed with the phase 3, where here the objective is obtain additional information about the effectiveness on clinical outcomes and evaluate the overall risk-benefit ratio in a demographically diverse sample. And factors to be identified are drug-to-disease interactions, drug-to-drug interactions, dosage intervals, risk-benefit information, efficacy and safety for subgroups. Data focus: laboratory data, efficacy, adverse events. Design: generally here we used to have randomized trials, well controlled, and 2 to 3 different arms, and here broader eligibility criteria. It will be conducted for several years according to the complexity of the clinical trial. It may be conducted for several years, and here the participants are nothing but individuals with target diseases, and the sample size is 100 to 1000. Once they are done with the phase 3, then they have to send their research information to the FDA as NDA, New Drug Application. Now, the FDA will review the new drug application, and if it was satisfied, then it will give the approval to release the drug or to market the drug. They will start marketing the drug. Once it has been released into the market, then they will proceed with the phase 4. Monitor ongoing safety in large populations and identify additional uses of agent that might be approved by the FDA. That means to be made it as a registered drug. Okay, ongoing studies. Factors that they will look for is epidemiological data, efficacy and safety within large and diverse populations, pharmacoeconomics. Data focuses efficacy, pharmacoeconomics, epidemiology, adverse events, design, uncontrolled but observational, ongoing, individual with target diseases as well as new age groups, genders, etc. etc. Okay, sample size is 1000. So they mainly concentrate in the phase 4 is to think about the long-term effects of the drug, and at the same time they used to compare their product with the already existing standard products regarding the effectiveness, price, everything. Okay. So generally this is the workflow when it comes to the drug development. As we have observed, from one phase to another phase, the number of participants are very high in number from one phase to another phase, and the time period is also very lengthy. If the researcher has to make a decision, or if the investigator has to make a decision regarding that particular chemical compound, there the need to have of SAS. So here the SAS is mainly used in phase 2 and phase 3 studies, and occasionally in phase 1 as well as phase 4, and in some situations even in the preclinical studies also. Whenever the research organization is conducting the trials, they will monitor everything. They will monitor everything, and through electronic data capture, we will call it as EDC. Through electronic data capture, they will monitor everything and they will capture every information into the databases. Earlier, all are used to maintain their data in open clinical database, but nowadays all are using Oracle clinical database. Okay. So the collection of data, maintaining the data, all this will be done by the CDMT, clinical data management. Okay. Of course, maximum all this work will be done by the clinical data management. They will collect the data, they will maintain the data, they will do the data entry, everything. While they were entering, at that time only they will enter it through case report forms, through CRF. Okay. But whatever may be the data that they have collected during the study, it will be called as a legacy data. Or when it comes to the normal terminology, you can call it as a raw data. In clinical terminology, we will call it as a legacy, or when it comes to the normal terminology, we will call it as a raw data. We know that whenever we are conducting the trial, our main motto here is to develop the drug and to release it. But whether our process is in a proper manner or not, whether we have to release the drug into the market or not, all that will be decided by the FDA. So we have the requirement to send all our research information to the FDA. But If the FDA has to look over our data, or if it has to give a decision regarding our data, it must, okay, accept our data. So how it will accept the data regarding that? It has given the guidelines. Why? Because every day FDA will get the data from different organizations. So for our trial only, as I said, we have different phases, we have different number of persons, and it will be conducted for different time period. So practically, if you look, okay, it will take several years, and we will get very huge amount of data. So maintaining the data and sending the data for a single drug itself will be a large process. But FDA is getting that kind of information every day from different organizations. So it is very difficult for the FDA to look over all the different data which came from different organizations. So it will take a lot of time for them to review every information and make a proper decision regarding that. That too, as it is related to the human health, okay, there must be more care should be taken. Because of that reason, FDA has given certain guidelines. Why? Because if they have not given the guidelines, each and every pharmaceutical industry will follow their own guidelines, and they will develop the data in their own pattern, and they will send it. So it will be difficult to understand in what pattern they have developed the data. Okay, in what pattern they were sending the data. It will be difficult for the FDA to understand that kind of information. That's why FDA and CDISC, Clinical Data Interchange Standard Consortium, and ICH, International Conference on Harmonization, they have given some GCP, good clinical practices. Okay, like that, different organizations has went with certain research, and they have given certain guidelines in what format we should send the data to the FDA. Now it has given a standard format for all organizations. Whatever may be the organization, when it is trying to send the data to the FDA, it must send the data according to the guidelines of FDA only. Okay, so there we need the CDISC, Clinical Data Standard Interchange. Okay, so when it comes to the, I already told you, when they were conducting the data and they were managed by the clinical data management team, and here they will present in semi-STTM format. When you want to send the data, especially the tabulations, to the FDA, then how we will prepare it, how we will manage it, what are the different domains that we have, all that will be specified by the CDISC in the STTM, Standard Data Tabulation Format. That means till now we have the legacy data, but if you want to send this information to the FDA, it must be standardized. So how we will standardize it? Okay, how we will categorize the data? All that will be explained in the STTM of CDISC. Maximum whatever may be the data that they are collecting, it is in the semi-STTM format. From that, the data will be sent to the STTM team. Okay, we used to have a separate team who will work on the STTM. Till this process, okay, maximum until this particular information, maximum the work will be done by the clinical data management only. But from STTM to semi-STTM to STTM, the actual work with the SAS will start. Here we will make the data standardized. If we have to send the data to the FDA, okay, according to their guidelines, okay, we will prepare the STTM tables. Of course, here we have different data sets or domains, like demographic table, adverse event table, okay, medical history, laboratory, okay, findings, exposure, disposition. Like that, we have different domains and data sets. So whatever may be the data that we got from CRF or from the clinical data management team, we will make that information into categorized, and we will make them standardized according to the CDC, and we will prepare the STTM tables, standard data tabulation model. Okay. So once we have prepared the STTM table, then that information will be sent to the Adam team. So we used to have a separate team who will work on the STTM, and we have a separate team who will work on the Adam data sets. But this is a chain. If the STTM tables are not existing, then we cannot prepare Adam data sets. The main purpose for Adam data sets is for analysis. Analysis data model. This is standard data tabulation model. This is analysis data model. Maximum, the analysis reports will be generated from the Adam. Of course, from the STTM tables also we used to generate the reports, but maximum general reports like listings and tables. But more analysis reports are prepared by the Adam. Because in Adam we have more detailed information. In STTM also we have detailed information, and we will make it standardized. But apart from STTM, here in Adam we used to have more detailed information. So in SAS we have a different team who will work on the STTM tables, and we have a separate team who will work on the Adam tables. And only for preparation we used to have a certain team. Okay, so there will be certain team who will prepare the STTM tables. We used to have a team which will prepare Adam data sets, but from them we used to have TLFs, tables, listings, and figures. We used to have a separate team who will generate the tables, listings, and figures, and then we have a separate team who will do the validation, or rather than that, the best word is verification. We have a separate team who will prepare the STTM tables and Adam tables. Once the tables are prepared, once the data sets are created, then from them we used to generate tables, listings, and figures. That means we will generate the report to send to the FDA. Once we have generated report, whether those reports are valid or not, whether the code is valid or not, whether the data is valid or not, whether the report is valid or not, we have validation as well as verification. Okay. So generally this is the work which we have with SAS in the clinical domain. But I can explain the same thing in another way. We have data accessing. We will access the data. Even though they were maintaining the data in the databases, maximum the data will come us to us as a file or in Access, MS Access. Maximum the data will be in Excel and Access. Even though they were maintaining the data in the Oracle clinical database, okay, majority of the data will come to us as a file or database. Maximum it will be in Excel or in Access database. We know that in SAS we have different techniques how to get the data from the Excel as well as Access. So once we got the data, here we will call it as a data set, or we will call it as a domain. Data set means normal SAS terminology, but domain is nothing but clinical terminology. As I said, we have different tables like demographic, adverse event, medical history, laboratory, okay, etc. And here we will do validation and cleansing. Remember that we will do it by the standard operating procedure, where it will be developed by the protocol. Protocol means it is nothing but analysis plan. Okay. Protocol means nothing but it is an analysis. Sorry. Protocol means it is nothing but the trial design. Okay. What is the objective of the trial, or what is the objective of the clinical study? Okay. What was the design? What was the sample size? How many number of participants has participated in the trial? Okay. How they have collected the data, how they have analyzed the data, how they have disposed of the chemical compound. Complete information. The complete information regarding the entire clinical trial will be done according to the protocol. Or in a simple way I can say, each and everything in the trial or during the trial must be conducted according to the protocol. We should not do anything apart from the protocol. Suppose if we got any situation where we used to do something out of the protocol, then we must give the protocol amendments. That means whatever may be the modifications that we are doing apart from the protocol, regarding that we have to specify the information, and according to that they have to conduct the trial. So from the protocol we used to prepare the standard operating procedures, which will help us to validate and clean the data. So in the protocol they said the persons who are having the age in between 18 to 65, only they have participated. So we have to verify whether the data is having all the persons with age in between 18 to 65. They said the blood pressure must be in this particular situation. Like that, they have given certain guidelines in the protocol. So according to that, we will prepare the standard operating procedures, and there we will do the validation as well as cleaning the data also. And once it is over, then we will prepare the analysis data sets, or we will categorize the data. Okay. We will make it categorized, or here we will make it even more standardized, or we will simply prepare the analysis data set which is used to do the analysis. And we will do the analysis according to the SAP, statistical analysis plan. Generally, whenever they were giving the data, of course in the protocol they will mention how we have to do the analysis, but in protocol we may not be having the complete detailed information. But when it comes to the statistical analysis plan, there we used to have more detailed, complete information. Actually what they were looking from the data. What is the primary endpoint, what is the secondary endpoint, and what they were looking for. All that information will be specified in the statistical analysis plan. Generally, the biostatisticians will prepare the statistical analysis plan, and according to that we will do the analysis, and then we will prepare the reports. Okay, it may be tables, listings, figures, whatever it may be. And finally, we do have validation. Validation means not only validating the report, we will validate the program, we will validate the report, we will validate the data, everything. So, yeah.
Hello?
Yeah.
This is one second. Actually what happened is my system is just not shut down, it's got, because I was not doing anything on it, it is just went blank. If I reboot the system, I don't know if my connection the connection might go. I can still listen to you, but I just tell you that.
Anyway, the thing is, we are done with the introduction. I will do one thing. I will send you the recording.
Okay.
Okay, I have just recorded this entire meeting. Okay, I will send you the recording. If you miss anything, just listen to the recording. Okay, then you can think about it.
I
Okay.
didn't miss anything. Just now it went. I think because I've taken the laptop to another room. It's not plugged in,
Okay.
so
Okay.
it's at sleep mode. It is moved automatically into that. I think.
Okay.
So typically when we're doing this, like I've been trying to actually study this for some time on my own, but I've been I find it a little actually too complicated in terms of like a lot of different like demographics or comments or whatever, lot of different things. So that's why I thought I'll take your help. So when you're going to be teaching me, it'll be like you'll be covering it. How would you be taking it? Taking the class basically.
Uh,
What and all
did you
will we be?
plug in?
I mean, I saw the content that you sent me. Uh,
Yeah. Can you watch? Did you plug in?
I have to reboot it once again. So give me a second.
Hello?
Yeah.
Did you log in?
I will just come. Yeah. I'm not able to see your screen.
No. Still, you're not here in the meeting.
I'm not in my just loading. Wait, wait. It's connected now. I mean,
Still you're not here. Yeah, now I got you. Check out, can you watch the desktop now?
yeah, I can see your this.
Okay. I have already sent you the contents. So,
Okay.
in this order only, whatever we have here, actually this is the content which you can find even in the SAS official website for clinical
Mm-hmm.
certification. Okay, same thing. So, whatever I have found there, I have got the same thing. Okay. So first we used to have some theoretical part regarding what are clinical trials, okay, what are the different phases, key roles, key organizations that we have, and what is SAP, and what are the CRF, annotated CRF, okay, and what are the guidelines, okay, regulatory
Okay.
requirements to submit the data to the FDA. We will discuss about all those, and
Mm-hmm.
clinical data structures, okay, um, what are the different classes that we have in the clinical trials or clinical trial data, what are the main principles of series, and what are ADAM, STTM, defined XML. We will think about all these things. Okay. Here, till this part, I will go with only theoretical explanation. Okay. I used to come with different
Okay.
presentations or with different documents, and I will explain them. Then importing it, that means actual programming part will start from here. Okay. Importing and exporting clinical trial. Okay. Of course, majority of the things we have already went through in the regular SAS. Okay. So I will just touch them and I will go a little bit deeper. Okay.
Okay.
So we will go with importing and exporting clinical trial data, managing clinical trial data, all this, and transforming. This is the main part or main heart of the training. Loops, arrays.
Okay.
Okay. Of course, we have seen them, but here we will go a little bit deep. LOCF, BOCF, all these things, functions and important functions and everything. and some of the important statistical procedures, okay, and macros, okay, and how proc report is used to do the thing. Okay. And before going to the validating clinical trial reporting data, here we will go with the preparing STTM and Adam. Already I have given explanation, but how we will prepare directly, we will go with examples. Okay, how to prepare Adam dataset and how to prepare STTM dataset. Okay, then we will go with the validating. And once we are done with this one, then I will show you a real-time project. If you are interested, I can give one or more also. So this will be the flow. Okay.
One more thing. One more question that I had is, like, after this, I will be able to, when I look at a person or if I take an interview, I'll be able to talk in those terms, or
Everything.
basic idea is that.
Not basic idea. We will go with examples. Okay.
Okay.
You will be able to suit for any position in the SAS, with SAS, when it comes to the clinical. It may be programmer, analyst, developer, any position with SAS. Okay. And you will be able to answer each and everything that you have mentioned in your resume. Okay. Each and every term. But because directly whatever we are trying to do in the real time, I am showing the same. Okay. Complete real time information. Okay.
Okay.
So you will not find any difficult to we will go with each and every term, and we will go with each and every example. And apart from that, if you found anything which you think it is related to the clinical trial, I will definitely explain even that one also. If I have not included anything that is present in the series. But because as I showed, whatever may be the contents that I have kept here, it is directly the SAS official URL only, not of my own. Okay, so there is no need to skip anything. Okay, so everything we will go with everything. Okay.
Great. Okay. When we start, Anandu?
Pardon?
When will we start? As in, like, we can start having the classes from tomorrow itself.
The thing is, as Thanksgiving is coming, I am having some long weekend plan. Can we start from first December, from Monday, freshly?
Yeah, that should be. Yeah.
Okay. Because I already have some other plans. Okay. So
No problem.
it's better to start from Monday. Okay.
Okay. Okay.
Okay then. Okay then. Bye.
Thanks. Bye bye.